Hello all, hope you're having a great day today. Today we are talking about the energy systems, the human energy systems. So here we go. Energy in the body is available for immediate use in the form of adenosine triphosphate or ATP. It is a complex molecule constructed with high energy bonds which when split by enzyme action can release energy rapidly for a number of body processes including muscle contraction. ATP is classified as a high energy compound and is stored in the tissues in small amounts. It is important to note that ATP is the immediate source of energy for all body functions, and the other energy stores are used to replenish ATP at varying rates. Myberg notes that muscle contraction is totally dependent on ATP, so the body has developed an intricate system to help replenish ATP as rapidly as needed. Another related high-energy phosphate compound, phosphocreatine or PCR, is also found in the tissues in small amounts. Although it cannot be used as an immediate source of energy, it can rapidly replenish ATP. ATP may also be formed from either carbohydrate, fat, or protein after those nutrients have undergone some complex biochemical changes in the body. This is the chemical equation for ATP and basically what it has gone through. So you need ADP or adenosine biphosphate plus phosphagen and energy. All three nutrients, carbohydrate, protein, and fat, may be used to form ATP, but carbohydrate and fat are the major sources via the aerobic metabolism of the Krebs cycle. Carbohydrate may be used to produce small amounts of ATP under anaerobic conditions, thus providing humans with the ability to produce energy rapidly without oxygen for relatively short periods of time. There are three energy systems that we utilize. One of those is ATP PCR system. We'll talk about it, as well as the lactic acid system and the oxygen system. Here we go. The ATP PCR system is also known as the phosphagen system because both adenosine triphosphate and phosphocreatine contain phosphates. ATP is the immediate source of energy for almost all body processes, including muscle contraction. This high energy compound stored in the muscles rapidly releases energy when an electrical impulse arrives in the muscle. ATP must be present for the muscles to contract. The body has a limited supply of ATP and must replace it rapidly if muscular work is to continue. The main purpose of every other energy system, including PCR, is to help regenerate ATP to enable muscle contraction to continue at the optimal desired rate. PCR is like I said, a high energy compound found in the muscle that helps to form ATP rapidly as ATP is used. Energy released when PCR splits is used to form ATP from ADP and phosphagen. PCR is also in short supply, but more than ATP, and has to be replenished if used. Phosphocreatine, or PCR, is stored in the muscle in limited amounts. It splits off the high energy bond release. Splitting requires the high energy bond release of energy, which can be used to rapidly synthesize ATP from ADP and phosphagen. ATP and PCR are called phosphagens and together represent the ATP and PCR energy system. This system is utilized primarily for quick, maximal exercises lasting about 1 to 6 seconds or more, such as sprinting. The ATP PCR system is critical to energy production because these phosphagens are in supply, particularly PCR in a given muscle. Hence, the phosphagens must be replaced, and this is the function of the other energy sources. Increasing the level of PCR in the muscle would provide a greater fuel reserve and possibly enhance the performance of brief, high-intensity exercise. Creatine supplementation is one way athletes are increasing muscle PCR in an effort to enhance performance. In summary, the value of the ATP PCR system is its ability to provide energy rapidly. For example, in sport events such as competitive weightlifting or sprinting 100 meters. Anaerobic power is a term often associated with the ATP PCR energy system. And this is just a diagram of what it looks like as it goes through the chemical equations. And you can see ATP plus creatine, and then creatine, that creates creatine phosphate, and breaks it down into ADP, which combined together create creatine kinase, and that splits off into creatine and ATP, which is used for muscle contraction. And another equation, this is, again, we're creating ATP, so ADP, or adenosine phosphate, and phosphagen plus energy create, and then creatine plus phosphagen and energy creates creatine phosphate and ADP. And on the flip side, you have creatine phosphate, which is creatine plus phosphate and energy, and then combine that with ADP and phosphagen and energy, and you've got ATP, which is the end result. The lactic acid system cannot be used directly as a source of energy for muscular contraction, but it can help replace ATP rapidly when necessary. If you are exercising at a high intensity level and need to replenish ATP rapidly, the next best source of energy, besides PCR, is glucose. Glucose may enter the muscle from the bloodstream or may be derived from the breakdown of glycogen stored in the muscle. The glucose molecule undergoes a series of reactions to eventually form ATP, a process called glycolysis. One of the major factors controlling the metabolic fate of glucose is the capacity of the mitochondria, which are cell organelles that need oxygen to process glucose to ATP. If the muscle cell mitochondria can process the available glucose, then adequate oxygen is assumed to be available, and this is known as aerobic glycolysis. Conversely, if the rate of glycolysis surpasses the capacity of mitochondrial oxidation to meet the energy demands of the exercise task or to maintain a high level of aerobic glycolysis, then insufficient ATP is formed and lactic acid is a byproduct of the process necessary to increase ATP production. This process has been referred to as anaerobic because of inadequate aerobic processing by the mitochondria, and the term anaerobic glycolysis has been used as a scientific term for the lactic acid energy system. It is used in sport events in which energy production is near maximal for 30 to 120 seconds, such as the 200 or 800 meter run. Anaerobic capacity is a term often associated with the lactic acid energy system. Muscle glycogen and glucose can break down without the utilization of oxygen. This process is called anaerobic glycolysis. ATP is produced rapidly, but lactic acid is the end product. Lactic acid may be a major cause of fatigue in the muscle. The lactic acid energy system is utilized primarily during exercise bouts of very high intensity, those conducted at maximal rates for about 30 to 120 seconds. The lactic acid system has the advantage of producing ATP rapidly. Its capacity is limited in comparison to aerobic glycolysis, for only about 5% of the total ATP production from muscle glycogen can be released. Moreover, the lactic acid produced as a byproduct may be associated with the onset of fatigue, as discussed later in the chapter. In brief, a prevailing hypothesis suggests that anaerobic glycolysis releases hydrogen ions, increasing the acidity within the muscle cell and disturbing the normal cell environment. The processes of energy release and muscle contraction in the muscle cell are controlled by enzymes, whose functions may be impaired by the increased acidity in the cell. However, it is important to note that the lactate present after loss of the hydrogen ions still has considerable energy content, which may be used by other tissues for energy or converted back into glucose in the liver. And this is basically what it looks like. Again, you've got blood glucose and muscle glycogen combining, the glycolysis reaction that occurs and creates ATP and pyruvate. And if you have oxygen, then you've got aerobic respiration. If you do not, then of course you've got lactic acid that buildup that occurs in the blood. And this is just the chemical equation associated with it. The third system is the oxygen system, also known as the oxidative or aerobic system. Aerobics is a term used by Dr. Kenneth Cooper in 1968 to describe a system of exercising that created an exercise revolution in this country. In essence, aerobic exercises are designed to stress the oxygen system and provide benefits for the heart and lungs. The oxygen system, like the lactic acid system, cannot be used directly as a source of energy for muscle contraction, but it does produce ATP in rather large quantities from other energy sources in the body. Muscle glycogen, liver glycogen, blood glucose, muscle triglycerides, blood-free fatty acids, and triglycerides, adipose cell triglycerides, and body protein all may be ultimate sources of energy for ATP production and subsequent muscle contraction. To do this, glycogen, fats, and protein must be present within the muscle cell or must enter the muscle cell as glucose, free fatty acids, or amino acids. Through a complex series of reactions, metabolic byproducts of carbohydrate, fat, or protein combine with oxygen to produce energy, carbon dioxide, and water. These reactions occur in the energy powerhouse of the cell and mitochondrion. The whole series of events of oxidative energy production primarily involves aerobic processing of carbohydrates and fats and small amounts of protein through the Krebs cycle and electron transfer system. The Krebs cycle and electron transfer system represent a highly structured array of enzymes designed to remove hydrogen, carbon dioxide, and electrons from substrates such as glucose. At different steps in this process, energy is released and ATP is formed, with most of the ATP produced during the electron transfer process. Although the rate of ATP is lower, the major advantage of the oxygen system over the other two energy systems is the production of large amounts of energy in the form of ATP, as you can see here depicted in the diagram. However, oxygen from the air we breathe must be delivered to the muscle cells deep in the body and enter the mitochondria to be used. This process may be adequate to handle mild and moderate levels of exercise, but may not be able to meet the demand of very strenuous exercise. The oxygen system is used primarily in sports emphasizing endurance, such as endurance runs, ranging from 3.1 miles to the 26.2 marathon and beyond. This chart depicts how far an individual could run based on using the total of each of these energy sources as the sole supply. The role of each of these energy stores during exercise is an important consideration. And in summary, the potential energy sources in the body include ATP and PCR, serum glucose, glycogen in the liver and muscle, serum free fatty acids, triglycerides in the muscles and in adipose tissue, and muscle protein. 3 human energy systems have been classified on the basis of their ability to release energy at different rates of speed. They are the ATP, PCR, lactic acid, and oxygen energy systems. The ATP-PCR system is for anaerobic power, lactic acid for anaerobic capacity, and aerobic glycolysis system for aerobic power, and then aerobic lipolysis system for aerobic capacity. The human energy systems for exercise can be classified as anaerobic or aerobic and can be subdivided into energy systems for power and capacity, as you can see here. With anaerobic power, that's primarily the ATP PCR system. Anaerobic capacity, the lactic acid energy system, also called anaerobic glycolysis. For aerobic power, you want aerobic glycolysis, and the aerobic capacity, aerobic lipolysis. Protein may be used as an energy source during exercise and has been referred to as aerobic proteolysis, but its contributions are considered minor, and it is not classified here as a separate energy system. And that is all we have for today. Thank you for joining me, and I look forward to talking with you soon. Have a great day.